Okay, so last we left off, we had talked about, well, we talked about a lot of stuff. So let's see. Can you tell me the example of the synthesis reaction that we had talked about?
Sodium and chlorine combining to make sodium chloride.
Okay, so that would be an example of a synthesis reaction. Yes, that's true. Yes, but what was the? We also talked about one specific one that happens in your body quite a bit with monomers and stuff like that.
Anabolism.
Okay, that process. And so, what does that require?
Two. Oh my gosh, I'm blanking. Two things to make a product.
So, is it two things making a product, or because catabolism? Is that building something or is that destroying something?
Oh, I said anabolism.
Anabolism. Oh, I thought you said catabolism.
Oh, no, because we were talking about synthesis. So,
So, but there is a specific type of synthesis reaction that we had talked about. What was it?
Dehydration synthesis?
Dehydration synthesis, right?
Yeah.
So that's going to be one of the most common ones that are going to be in the body. And then the opposite of dehydration synthesis would be what?
Hydrolysis.
Hydrolysis. And that's using what?
Using water to break things down.
Okay. Good. You guys are staying on top of things. I love it. Makes it easy. Okay, so let's talk about, we did talk about the different types of bonds. Okay, what are the three different types of bonds?
Ionic, covalent, and hydrogen.
Okay. So which one are kind of strong bonds and which one are more like an interaction?
The covalent's the stronger one. And you want the reaction or the easier to pull apart?
So which one is the easiest to pull apart? Who's the weakest?
Hydrogen.
Okay. So ionic bond. So what is the difference between an ionic bond and a covalent bond?
the ionic gives their electrons away while the covalent shares.
Okay. So ionic bond is about give and take. So one is taking an electron and one is giving an electron, right? And then those charges interact. And then with the covalent, you're sharing. So what type of sharings do we have?
Polar and non-polar.
Polar and non-polar. Okay, good job.
Thanks.
Impressive. Okay, so you guys ready to get started?
Yeah.
If I can find my screen.
Yes.
Okay, so just a quick update. I had submitted, I had put up, you have your next reading assignment already up. So for your reading assignment, your next reading assignment, what I want you guys to do is provide me with a PDF of flashcards that you're making. So you have to give me at least 10 terms that you've looked up and found the definition to. On one side, you'll have the term. On the other side, you'll have the definition. And then you're going to submit a PDF with 10 flashcards on it. So it could be a PDF or it could be a JPEG. Okay.
With terms from chapter 1 and 2, right?
No, chapters 3 and 4.
3 and 4.
Yeah. So you guys should have already read chapters 1 and 2, right? You guys know this like the back of your hand already. But if you want to make flashcards for that too, you can do that as well. But I didn't give you guys that assignment. And so you guys remember on Friday, you're going to have a quiz that's going to open up your chapter quizzes. So that will be on chapters 1 and chapters 2. So everything that we've covered so far is fair game.
When does that quiz close?
It'll close on Monday night. So it'll be open from Friday to Monday night.
Okay.
So don't wait till the last minute to do it because stuff always happens. Okay, so can you guys see my screen?
Yes.
Oh my other business. Okay, so when we talk about dissociation of sodium chloride, so sodium chloride, when it's interacting, when there's no water around or anything like that, it is a very strong interaction, but it's mainly done by electrostatic interaction, plus and minus coming together. Okay, now water actually gets between them, right? So what water does, because water is polar, it makes that partial negative and that partial positive. You guys see that partial negative for around the oxygen, and the partial positive around the hydrogen, right? So water will actually surround that sodium, right? And then so all of the negative, those partial negatives will surround the sodium ion, the sodium plus ion. And then all of the hydrogens will surround the chlorine ion, right? So it'll separate the two. It's kind of like a home wrecker, right? So you have sodium and chloride that are together. They're nice little happy couple. And then water comes along, and so water says to sodium, "Hey, baby, you don't need him," right? And then sodium says, "Oh, water is so hot." Woo, woo. And then as soon as sodium turns around, water goes to chlorine. "Hey, you don't need her," right? And then so water is playing both of them. It separates them and causes them to dissociate. And because it dissociates, it's able to be sustained in water, right? So if we put salt in water, what happens?
Usually dissolves.
It dissolves, right? Unless you have a whole bunch of salt. And then that just means that you don't have enough water molecules to surround the sodium and the chloride, right? So you basically separate the two so that you have just the ionic forms surrounded in water. Make sense?
Yeah.
So water does this with a lot of things, right? So it'll actually do this with other ionic compounds as well, too. And so how many of you guys drink alcohol?
I
You
do.
don't drink alcohol? Good,
I do.
good. You can just give it all to me. That's fine. So anyway, so if you think about, we put ice in our alcoholic drink, right? And so if we just sit around and just talk a lot, doesn't that ice melt? And then eventually your alcoholic beverage gets watered down. Is there a separation between that water and the alcoholic beverage, or does it blend?
It
There's a
blends partially.
separation.
Sometimes it sits on top.
Well, it also depends on what else is in it. But let's say over time, if you stir it, it'll just mix nicely, right? And you
Yeah.
can't tell the difference between it. So in that case, we have CH2CH, say C2H5OH. So this is ethanol. So let me show you what it kind of looks like. I'm going to draw a really crude structure. I won't do the bunny ears and all that stuff for this guy. So it's carbon, carbon. Okay, so because this guy has this nice little OH, it's able to interact with water. So again, you'll have water, which is, so this guy here is going to be your partial negative. This guy's going to be your partial positive. This guy's going to be partial positive, and this guy's going to be partial negative, right? So then the partial negative is going to interact with this hydrogen. So it's going to form a hydrogen bond. So the water is going to come along and orient itself like this. And then the hydrogens are going to orient itself towards this oxygen and form a hydrogen bond. So then it keeps the alcohol in solution as well. Where these guys, if it was a long enough tail, these guys would be kind of hydrophobic, right? Carbons are kind of non-polar, so they're not going to want to interact because they don't have any charges associated with it. But since it's a very short molecule, it's going to take advantage of this. But if we had a long carbon chain, right, only this part would be able to interact, this part wouldn't. And so the longer the chain is, the less soluble it would be in water. Does that make sense? Okay, so for instance, we have fat. Fats are triglycerides, you guys, and we will be talking about this in a minute here. So, and then I'll show you the image of fats. So on one end, it has a charge, but on the other end, it's just long chains of carbons, right? So if I put fat in water, you guys ever put oil in water?
Put what in water?
Oil.
Yes,
Okay, so when you put oil in water, what happens?
it separates.
It separates. Okay, why is that?
Isn't one more dense than the other?
No, that's not the reason why. That's the reason why one floats on top of the other. Yes,
Oh,
but that isn't the reason why it separates.
I'm not sure.
Okay, so in the case of that, you have these really long hydrophobic, they're called hydrophobic chains, right, of carbon. Hydrophobic also is non-polar. They're long non-polar chains of carbon, so they don't interact because they're not based upon those charges. Okay, so all of those guys, since they're all hydrophobic, they're going to interact with each other. They're going to want to stay away from water since they don't have any polar moieties to cause them to interact. Okay, and I'll show you an image of that here in a second. Okay, so where sodium is all charged, it's able to be dissolved in water. And alcohol, as this charged in, it's able to dissolve in water. Okay, so let's see here. So have you guys heard of acid and bases? Not acid as the drug, but
Yes.
okay, so you guys know a couple of acids?
Yeah. Lemon juice.
Lemon juice. Okay, so lemon juice is an example of an acid. So with acid and bases, acids are, they basically are compounds that are able to dissociate and provide what we call the hydronium ion or a hydrogen ion. So a hydrogen ion, so let me give you an example. We have hydrochloric acid. Okay, so when it's in water, it dissolves and it dissociates. So what happens is this guy becomes H+. And we use AQ stands for aqueous. It just reminds me that it's a water solution. Plus Cl minus. So it separates into its ionic compound. So you remember, just like ions, acids are able to dissociate into its ionic parts. Okay, so bases, on the other hand, also dissociates into ionic components. But in this case, you have, typically it'll be like a metal and the hydroxide ion or the hydroxyl ion, what they call it in your book. So it'll separate into A plus, AQ, plus OH. So we come across a lot of acids in our body. So your stomach, right? So your stomach is composed of acid. Have you guys heard of acid reflux?
Yes.
Okay, so what happens in that context is that you have pumps in your cells that are in your lining of your stomach, is that they're pushing out a whole bunch of H pluses, right? And so you have a lot of H pluses available, and that gets in solution in your stomach, and then it just comes back up. And so it burns like crazy, and you get really bad heartburn and all of that, and it starts burning your esophagus because you have a lot of acid in your stomach because you're producing a lot of excess acid because your stomach is unhappy. Okay. And so normally your stomach uses acids to help break down food. And so it uses a lot of the acids there and do it. And so often what we'll do is that we'll get stuff that is kind of a little bit basic to sit there and try to neutralize it, like an antacid, right? So an antacid basically has some components to kind of help neutralize that base. If you guys take Tums or something like that, you get a little bit of heartburn. No, you don't?
Yes. Yeah, you do.
Or baking soda,
No.
you know? Yeah. So both of those will work with that little bit of heartburn. You know, just take a spoon of baking soda, right? Okay. So now, when you have a lot of H plus is around, your pH basically stands for, it's the minus log equals minus log the concentration. You guys know what concentration means?
No.
Okay, so concentration is how many of those molecules that you have in solution, right? So H plus, so if it's low concentration, or we call it dilute, that means you have very little in there. If it's high concentrated, or we call it high concentration, or we call it concentrated, then there means there's a whole bunch. How many of you guys have made orange from the little can orange? Anybody?
Yes.
Right. So isn't that called concentrated orange juice? Right. So all they've done in that case is removed out some of the water. And so they pack it up and they remove out some of the water that would normally be in orange juice to make it more orange juice molecules in there, right? And then, so you just plop that into water and then you make it dilute, right? So you go from a concentrated form of it to a diluted form of it. Does that make sense?
Yeah.
Okay.
Yes.
Okay, so we're looking at the concentration of the H plus ion, right? And so this will allow me to calculate the pH. So the more H pluses I have, right, the lower the pH. The less H pluses I have, the higher the pH, okay? And then in the case of a base, you're looking at the amount of hydroxide, or the hydroxyl ion, which is a negative charge. So in that case, you're looking, it would be your POH, which would be the log of that. Oops. Minus log of the concentration of the minus OH. Okay, this is just like a little extra information, right? So, and then that'll let us know the POH of it as well. So basically we have this nice, beautiful scale. And because we know that, well, I should say, I know, you guys probably don't know. So this scale is kind of based upon water. Water is neutral. Water is considered to be pH 7. So at pH 7, that means that you have the same number of H pluses and the same number of OHs, right? They're equal to each other. So this is the way we would write a chemical equation, just so you know. So water can break down into these ionic components. Very little of it does. So it has what we call a pH of 7, pH of 7 when it's neutral. So that means that you have a balance between your pH and your OH. Both of those are equal to 7 for water. Okay, so if something is more basic, then your pH is going to be higher. So it'll be greater than 7. And then if it's more acidic, it's going to be lower than 7. So and I'll show you a slide. Be clear. It's not the prettiest slide. So pH of 7, we're neutral. So blood is a little bit above, or should be just a little bit above pH of 7. So our blood is a little basic. Okay. And then acids are, oops, sorry, that's bases, sorry. Acids are below, lower than 7. And then bases are going to be higher than 7. Okay, so these are examples. Remember I told you baking soda? Well, baking soda has a pH of 9.5. So if you have a lot of acid in your stomach, you're basically just adding a little bit of base. Okay. Ammonia has a pH of 10.5. I'm not saying drink ammonia. Don't drink ammonia. Don't drink bleach. Don't drink any of those. Baking soda, you know, we use baking soda to bake. It's okay to do a baking soda because it's just a little bit off. Okay. So do you guys drink coffee?
Yeah.
Yes, I have mine right here. And that's part of the reason why I have such bad acid reflux is because I drink so much damn coffee, trying to keep myself going, right? So that is a pH of 5, right? So I often will need a little bit of baking soda during the day to kind of help balance me out. Okay, sodas are about a pH of 2 to 2.5. Lemon juice, pH of 2. Vinegar, pH of 2. Battery acid, don't drink battery acid. Okay.
What about carbonated, just regular carbonated water like LaCroix and stuff? Does that have any acidic properties to it?
So carbonated sodas, what happens in the case of that carbonation or carbonated waters? So they also do form what is carbonic acid. So it'll be slightly acidic, but just a little bit. Okay, because it forms what we call an intermediate acid, but it'll be like something like 6, 5, 6 even, something like that, compared to like regular soda, which has phosphoric acid, which is a pretty strong acid. It's a little bit of phosphoric acid in it, and then that guy makes it a lot more acidic.
And the phosphoric acid is for what? For preservatives, or what are they added in there for?
It does two purposes. Yes, it's partially for the preservatives, and it also gives it a little bit of its flavor. So all sodas basically have that in it. So, okay. Any other questions? Okay, so that's why if any of you guys are thinking about being dental hygienists and stuff like that, the dentist always says, don't drink soda, right? The reason why they tell you don't drink soda, because it has phosphoric acid, and that acid actually starts removing some of the enamel off the teeth. But it tastes so damn good and it's hard to stop, right? Yeah. So LaCroix doesn't do as much damage to you, so they usually don't complain too much about that. So, although it is slightly acidic. Okay, so let's talk about, so there are what we call bio molecules, right? Or bio macromolecules. So these are the kind of the building blocks of our bodies. So we have a couple of them. One of them are called saccharides. So we have monosaccharides and polysaccharides, or sugars, right? You guys have heard of sugar before, right?
Yeah.
It's like a drug. How many of you guys eat those muffins? Or cupcakes, you know, right? We love our cupcakes, we love our muffins, tasty, right? We love them so much because they have a lot of sugar in them. So these are examples of what we call monosaccharides. Monosaccharides are a single sugar, mono meaning one, right? So it's made up of a single sugar chain. So glucose, fructose, galactose, and then in case of DNA and stuff like that, we have deoxyribose and just ribose. So these are single sugar chain. We're going to see these throughout our bodies. These guys are hexoses. They are 6 chain sugars. So that's 6 carbon sugars. So that's each of these corners represents carbon. That's 1, 2, 3. And then this guy is a carbon, 4, 5. And then that's your last carbon, 6. So it has 6 carbons in this, and it's called hexose, hex meaning 6, because it's a 6-ring chain or 6-ring carbon. So 1, 2, 3, 4, 5, 6. It has 6 rings to it. This guy is a 5-ring carbon, but it's still considered a hexose because it has a 6 carbons associated with it, where this guy has 4, 5, 6. So they all have 6. I'm sorry. So this guy actually should be not a hexose. It should be a pentose. Okay. And then galactose is also a 6-ring carbon. So if you guys take a look at glucose and galactose, right? Chemically, and fructose, they're chemically the same C6H12O6. They're all the same chemical structure. The only thing difference between, let's say, glucose and galactose is we have this guy flipped. And this guy flipped, right? So, do you see the difference?
So what makes fructose what do you call it, a pentose?
It's a
I mean, why-
pentose
why
because
is
it's
it-
5
why
rings.
is-
It's
why is
five,
it structured?
5. It has a
Yeah,
5 ring.
I know 5, but so like you were pointing, it has the same number of carbons, and you said that each corner represents a carbon, right? So why does it have 5 instead of 6 if it's a pent?
So, so usually, let me correct myself. So you are correct. I made a mistake. It's considered an hexose, right? But it's a 5 ring carbon instead of a 6 ring carbon. It has 6 carbons, 1, 2, 3, 4, 5, 6, where our deoxyribose is a 5 carbon, right? So that's 1, 2, 3, 4, 5. Okay, so it's still pentose because it has 5, but I should have said fructose is a 5 ring carbon, even though it's a hexose. It does have 6. Okay, so these guys, they're all of these guys here have the same chemical formula, C6H12O6, but structurally they're different. So these guys are considered to be isomers of each other. Iso meaning same, mers meaning that they're same formula, right? But structurally they're different, and they're going to behave differently in your body. So you guys know fructose comes primarily from where?
Fructose what?
Fructose comes primarily from where?
Fruit.
Fruit. Okay. And glucose comes from a lot of plants like corn, you know, things like that. It's high in starches, and in sweet corn particularly is high in glucose. Okay. So in the case of these guys behave differently in your body, right? So this is the ideal form. Fructose has to be modified to be changed into glucose. So it takes a little extra to sit there and to utilize fructose. So you use a little more energy for the utilization of fructose. So that's also why doctors say, hey, eat fruit, right?
So then it has
a little bit more.
does that mean that it has less influence on insulin fluctuations? So like glucose hits your bloodstream a lot faster, right? So then
That's right.
get insulin spike.
That's right. Where it would take fructose a little longer to hit your bloodstream because it has to be modified to glucose, then it can be utilized. Okay. And we will talk a little bit about the enzymes that are responsible for that, but a little bit. Are you guys good so far? Okay. So these are going to be the most 5 important monosaccharides in your body. So you want to be familiar with those. Okay. So then we also have what we call disaccharides, right? So sucrose, lactose, maltose. So sucrose is a combination of glucose and fructose that have come together. So sucrose is table sugar. So this is the sugar that we use to bake our muffins. So our body has to break this down, you know, and use the glucose, and then it'll use the fructose. So it'll use up all the glucose first. There's enzymes that'll separate. So what is that process of breaking down 2 monomers of sugar? So we have a sugar molecule that's together, and we break it down. We use what again? What?
Dissociation.
Well, yeah, it'll be dissociation, but there's a process, a specific process that we use. What do you think we're going to use?
Dehydration synthesis?
Dehydration synthesis. Yes, so we're going to use, well, are we going to use dehydration synthesis? That's what we use to make it. But to break it down, are we going to use dehydration synthesis?
Hydrolysis?
We're going to use hydrolysis. Hydrolysis would break it down to allow us to sit there and utilize it. Dehydration synthesis is what we would use to make it. So all of these guys are formed. So we take glucose and fructose, and it comes together to make sucrose. So all of these guys are formed by dehydration synthesis. Does that make sense? So like plants make sucrose, like what's it called? European sugar from Hawaii. What is the little sugar sticks? Can't even think of it now. What was that?
These
The
sticks?
pixie sticks.
Not candy.
Oh, okay.
Sugar cane?
Sugar cane. Thank you. Sugar cane. Yes. So sugar cane, right? So a lot of sucrose comes from sugar cane or beets, right? So those are your two common places of sugar, are beets and sugar cane. Have you guys eaten beets before?
Yeah.
Tastes like blood, right?
Oh,
So beets is a high source of sugar, and also sugar cane is a high source of sugar. So sugar cane is known for making high levels of sucrose. Okay, so what it does, it takes that glucose that it generates, and it also has fructose, and it binds it together by dehydration synthesis to make sucrose. Okay, so lactose, where do we find most of our lactose at?
would that be milk?
Milk, that's right, milk. Dairy, exactly. So in the case of milk, in our bodies, in the case of women, and then also cows, same place, in female cows, they make milk, right? And so in that case, you basically have galactose and glucose that are being bound together to make lactose. So galactose and glucose, they come together to make our galactose. And then we have what we call maltose. Maltose is just two sugar chains. Have you guys have like a malted shake or something like that?
Delicious.
Delicious, right? So maltose is a combination of two glucose molecules coming together. Okay, and so all of those are synthesized, made, each of these disaccharides are made by dehydration synthesis. So, and then our body breaks those down by what again?
Hydrolysis.
Hydrolysis. Okay, so amylase and amylpectin, those are examples of starches, right? You guys have heard of starch before.
Yes.
Okay, so those are starches, amylase and amylpectin. So those are your two forms of starches. So that's what we're going to find in our potatoes and in our corn. So our wheat, you know, so those are examples of that. So all those these guys are called polysaccharides. So they basically just have long chains of sugars that are connected together, right? And so amylpectin has branching sugars. Amylose basically it's just a long single chain of sugars. So that's the distinction between the two. Okay. So our bodies, how many of you guys work out, build muscle?
Sometimes.
Sometimes. Okay. So when we build muscle, we often will eat a lot and stuff like that to sit there and utilize what we call glycogen. We also build glycogen. Glycogen is basically long chains, long branching chains of glucose. So that have been combined together, covalently linked. Okay, so our muscles have glycogen in them. So that's what our muscles kind of use as reserves for food, right? So we store a lot of in our muscles, we store a lot of glycogen. So when we're working out, we build a lot of muscle. We basically establish a lot of places for storage of glycogen. Okay. Now, cellulose, not the stuff not the veiny stuff on your legs. Cellulose is basically fiber, right? So you find a lot of cellulose in basically bark, things like that. So the reason why we eat a lot of veggies is so that we get our fiber. So we're trying to get that cellulose. So what is the benefit of cellulose? Well, cellulose is, our body can't break it down, but it just kind of passes through us. So if we eat enough of that, it just helps us poop really well, right? So we need that fiber for pooping, right? Are you guys with me so far?
Yes.
Okay, so those are all examples of saccharides. So we have monosaccharides, we have disaccharides, and we have polysaccharides. Okay, so those are all of our sugars. So, and sugars we know, or you guys should know, they're really good at storing energy, right? So all of those carbon rings and everything like that have a lot of energy stored in them. So those bonds stores that energy that we're able to utilize later on. So we use starches, like plants use starch for food storage. We use starches to eat. So we break down starches to provide us glucose so that we can make ATP. So at the end of the day, we get ATP out of those by processing. We'll talk about that later. Are you with me? Okay. So saccharides are one of the major building blocks of our body. The next major building blocks are basically fats. So in this case, we have triglycerides. Okay, so they're called long fatty acid chains. So basically, the fatty acid chains are just long carbon chains. So they're just really long carbon chains. At the end of our carbon chain is what we call the triglyceride. So this together, all together, is considered the triglyceride. So at the end, this is glycerol. So glycerol, and glycerol is just basically it's a 3-chain carbon that has OHs at the end of it. And then so these OHs basically we basically remove water from each of these guys. So that process is called what again?
To my hydrolysis again?
Is it hydrolysis if we're removing water?
Oh, dehydration.
Dehydration. So it's dehydration synthesis. So through dehydration synthesis, we combine the glycerol to make a triglyceride. So the triglyceride is what fat is. That's our fat, neutral fat. So in this case, it's not charged. It's neutral. And then we also get free waters produced. Okay. How you feeling so far? Not too bad. And so these are long fatty acid chains. You start from glycerol. These are just your fatty acid chains. They come together to make your triglyceride. Okay. Okay. So when we start talking about fats, right, or fatty acids, there are 2 types of fatty acids. One is called saturated. Now, when we make the term saturated, it basically means it's saturated with hydrogens, meaning that there are no double bonds or triple bonds or anything like that. You just have all single bonds. These guys represent single bonds. Double bonds is when you're sharing 4 electrons between 2 molecules. So remember we talked about with covalent bonds, they're sharing electrons. So you can share 2 electrons, you can share 4 electrons, or you can share 6 electrons. 6 electrons means okay. If you're sharing 4 electrons, it's a double bond. If you're sharing 2 electrons, it's a single bond. So these guys up here are all single bonds. So this guy is considered to be saturated because all you have here on this side are single bonds. Okay. This guy is considered to be unsaturated because you have at least 1 double bond or triple bond. So you guys see the double bond right here? Okay. So anytime that you have a double bond or a triple bond, it causes a kink in the structure, where if you have all single bonds, you don't have a kink in the structure. Does that make sense? And that kink is because you have to share those electrons.
So then why is too much saturated fat bad for you? Why is it bad in the body?
So too much fat in general is bad for you, but particularly saturated fats, because the way you go about processing it, right? They're easy to process. They're easy to process because you don't have as many kink structures, but a lot of times you have what we call branching structures, right? So you'll have a single bond coming off the single bond off. So you could have carbons, long carbon chains coming off of here forming branches. And so what happens is that so your body needs fats in general, right? And primarily it uses a combination of saturated fats and non-saturated fats for membrane structures. And we're going to talk about that here in a minute, call it phospholipids. But if you have too much fat, what happens is that you get the fats starting to interact with each other. And if you have long chains of fats, it makes it easier for it to interact with each other, so you get those clogs. So it starts clogging up areas because they're really easy to interact. The kink causes like structural changes to make it easier for it to be broken up, where the saturated fats all together, they're just layers of fat on top of fat that allows it to just kind of sit on top of each other. Does that make sense?
Yeah.
So where, like, it's like this. So if you have a whole bunch of linear, get my pen, a whole bunch of linear structures, right? These guys. So they're just going to lay on top of each other because they interact by what we call these Feneral Forces. And since fats like to interact with fats, they're able to do it. Where if you have the kinks that are happening like this, and then you have another one that's kind of, well, you just have this little point of interaction. So this guy makes easy clogs, right? So just get fat clogged on top of each other, right? And then where this guy, it's easy to be broken down. So enough water could come in there and start to kind of separate the two, cause them to go separate because of the kinks. Okay. Good question though. Okay, so the next thing is phospholipids. Now, the phospholipids is kind of composed of two components. It has what we call a polar head, meaning that it's able to interact with water, and it forms those charges, right? Slightly charged associated with it. And then it has nonpolar tails. The nonpolar tails are long carbon chains that are hydrophobic. They're nonpolar, right? So they don't want to interact with water, hence hydrophobic. Okay, so the membrane kind of associates itself based upon its polar head and hydrophobic tails. Okay, so the way it works is, and so you'll often see it drawn like this, right? Polar head, hydrophobic tails. Okay, so polar. So if it has a polar head, is it going to want to interact with water, or is it going to want to interact with fat? Polar. Water. So this guy here, polar head faces the water. So your cell membrane, this here represents, it's going to be your cell, right? So you have a double membrane, right? So it's composed of these little guys. So you basically have all of these guys here forming these, your heads, and then here's your water. This is like your blood and all that stuff. This is your water out here. So these guys are going to be facing the water, and then the tails are going to be on the inside, and then inside your cell, you also have water, right? So then on the other side, you're going to have these guys as well, right? And so your membrane basically uses the hydrophobic parts of it to form like a little barrier, right? So that nothing can get in. It protects the water from getting in that way. So you can't get water in through this because of the hydrophobic tails, right? Your polar heads will interact with the water so that it stays kind of sustained in the solution and is able to interact, but water can't flow through easily. So we'll need a way of making water flow through. And often what we use to allow water to flow through are called channels. Okay, so we have what we call aquacorins. Aqua meaning water, corins meaning pore, right? So it's like water channels that allow water flowing in and out of the cell. And these guys are kind of controlled because we don't want too much water in the cell, right? Because then the cell will get too big and explode. We don't want too little water in the cell. So it's kind of controlled. It's regulated, okay? Okay, so Phospholipids, phospholipid. Lipid means fat. So this family is called the lipid family. So we have the triglycerides is a lipid fat family, right? So phospho meaning it has a phosphate on one side, and then it has the lipid on the other side. So this guy is your phosphate, right? This guy is your lipid. And then on the other side, these guys are various specific chains that makes it distinct, because there's various different types of phospholipids that are used for different membranes in your in the cell itself. So like the outer membrane is composed of a percentage of different type of phospholipids. Okay, so we have phospholipids, we have triglycerides. Okay, and then the other thing that we have are steroids. You guys heard of steroids, right? So that's like cholesterol. Cholesterol is a steroid, right? And I'm not talking about the synthetic steroids that that you pump yourself up to get bulkier. Okay. So cholesterol is an example, and then we have let's see. Let me erase some of this here. Okay, so here we have cholesterol, and then we have read. I see it at the bottom. Oh, here we go. Prostaglandin. So these are two examples of steroids that we basically will see in our body, right? And so this guy, you can see the ring structure here, right? I'm sorry. And sorry, you have steroids and prostaglandin. Right? So again, carbon chains with like a carboxylic acid in, right? And so we'll basically see these guys make up structures in the membrane itself. So like cholesterol actually sits on the membrane structure. And then we have good cholesterol, and we have bad cholesterols and stuff like that. We'll talk a little bit about those and a little bit more about those and how these guys act in terms of mobility of the membrane, because they're really important for that aspect. And how rigid your membrane will be. Okay. So we have lipids, we have saccharides, and then we have proteins, right? So that's our third building block. So proteins are made up of amino acids. So the next one would be proteins, and that is our amino acids. So this is the general structure of an amino acid. It's called an amino acid because it has an amino group. So this here represents your amino group. Nitrogen, anything that would have nitrogen, anytime you see AMIN, that means it has nitrogen in it. Okay, so amino means that you have N and the OH here, and it's usually going to be connected to like something else. So this is our amine group. This guy is considered our side chain. So do you guys know how many amino acids you guys have in your body?
Not really.
Okay, so you guys have 20 amino acids in your body, right? So they all differ by the side chain. The amine group is going to be the same, and the carboxylic, this is basically a carboxylic acid group or carboxyl group. This guy are going to be the same. The only thing that is going to change is this little R. So the R represents just that variability of chains that you get. So depending on the amino acid, it's going to have a different R group. Okay, so these are the building blocks for proteins. So do you guys take amino acids? How many of you guys take lysine in the morning, or you take a multivitamin? You guys take vitamins?
BCAAs.
Yes.
Yep. So if you take a look at your
Acids.
vitamins, it has a listing of all of the amino acids. So your body can make some of those amino acids, but there's certain ones that it can't make or has a little trouble making. So often in our vitamins, we have the building blocks so that we can have enough to sit there and sustain ourselves, right? And then a lot of the vitamins, a lot of the amino acids we get from meat or from proteins. So what we'll do is we'll eat up meat, or we'll break down the amino acids of that protein and utilize that for our bodies.
So
Okay.
our body like uses amino acids throughout the day. So like you have 20 all the time, or you like kind of fluctuate?
Okay, so the amino acids, you have all 20 amino acids available, right? Well,
Right.
our body can only make, so our body can make some of those amino acids, but there are a few of them that we can't make. So we have to get those from other sources. So we'll get it from like the cow that we ate, or we'll get it from vitamins, we'll get it from other things, right? So our body is able to make most of them, but it can't make them, okay?
Okay.
And so there are certain ones that we have to have. And the ratio of those amino acids vary on protein to protein. So it really just depends on the protein and the protein sequence, which would be the amino acid sequence of that particular protein. Does that make sense?
Yeah.
So for instance, DNA, when we talk about DNA is not a protein. DNA is it's one of those sugars, so it's nucleic acid, but it's also it has a sugar on it, so it's considered part of that sugar family. But we're going to talk about it independently because it's the building block of our genetic code, right? So DNA has 4 different things that we're looking at. Amino acids have 20, so those 20 are going to be interchanged to make proteins different from each other, right? Proteins can be different sizes. They're not all going to be exactly the same size, but the building block of those proteins, the monomers for those proteins, are those different amino acids. Does that make sense? So
So
you guys play with Legos. Ever play with
Yes. Yes, I
Okay.
do.
So it's kind of like Legos. So not all of your Legos are the same. Aren't they different? And that kind of gives you your kink, or that makes it straight, or, you know, and so your amino acids are kind of like different Legos. So you're putting the Legos together to make a different structure for each protein. Like, so for instance, if we were talking about insulin is a protein, right? And so insulin does a particular job. So it's going to have a different structure than, let's say, the aquacorins that are in the pores, right? And so depending on the different structure it has, it's going to have different properties and different responsibilities. And if you change, sometimes if you change a single amino acid, it can cause a protein not to work at all. So, like for instance, have you guys heard of sickle cell anemia?
I've heard of
Sickle cell anemia is a result of a change of a single amino acid. You change one amino acid, it causes your blood cell to be sickle shaped instead of being round. Just the change of one amino acid causes that. Okay. So that's an example. So it's important that we understand that it's certain structures that causes those mutations. And so sometimes if you have not functioning insulin is a result of single mutation change, or sometimes as a result of other things, or multiple changes. Okay. Are you with me so far? Okay. So by dehydration synthesis, we can actually build up our proteins, right? Or we can make what we call a peptide bond. It's called a peptide. Peptide kind of is that abbreviation for proteins. So you have peptides, and then we have polypeptides, and then we'll have proteins. So it's called a peptide when you have more than one amino acid together, it becomes a peptide, right? And then we have multiple amino acids together, it's called a polypeptide. And then we have a complete protein together, that's when it's already cut and it's really active and everything is working. That's our protein. Proteins are made up of multiple amino acids that are combined together by dehydration synthesis, that are bonded together by dehydration synthesis. And just that process of putting 2 amino acids together, we call it just peptide bonds. Okay, so we specify peptide bonds for that. Okay, so peptides or polypeptides. So we have different, how do I say it? So we have different types. Okay, so in a protein, a final protein is, it forms a certain shape. If it doesn't form that certain shape or flexible shape that it has, it won't function. So if we look at the peptide sequence, we have what we call the primary sequence. It's just the covalently bonded amino acids together. So that's considered our primary sequence or our primary structure. So it's just the covalently bonded structure. Then that polypeptide will then form what we call a secondary structure. So it's going to either be an alpha helix, meaning that it's going to form like a circle, something along those lines. So here, that circle, an alpha helix, like a curl, right? So that's what this kind of represents, kind of like a curl. And these guys are based upon hydrogen bonding or just interactions between the amino acids, the different amino acids in the primary chain. Or it's going to form what we call a beta sheet. So this guy represents a beta sheet. So here, this is your first amino acids, and it just kind of forms like a nice little sheet that's kind of folded up, right? So, and then sometimes you'll have a protein that has both alpha helix and beta sheets. So you're not going to have one or the other. You could have an area where it's an alpha helix, and then another area where it'll be a beta sheet. Okay, so this guy, the interactions between each of the different amino acids to sit there and form that secondary structure, that's called our secondary structure, right? And then we have what we call a tertiary structure. So in our tertiary structure, basically we get some weird folding that goes on. So you have regions of that polypeptide that are interacting with each other to form like some type of structure. That's your tertiary structure. And your quaternary structure is basically when you have other subunits coming in and interacting. So if you have multiple subunits that come in and interact together, that's going to be your tertiary structure. Okay, so let me see. So primary structure is just the amino acid sequence. It is like, first we have a glycine, we have lysine, we have glutamate, so that would be your tertiary structure. Your alpha helix is those amino acids that are combined together covalently, interacting with each other, right? Forming alpha helix and beta sheets. Tertiary structure, that's going to be folding. So it's just kind of like me taking this guy and just folding it up, right? Like this, okay? And then quaternary structure is me taking another protein. So let's say this is my other protein and this guy, and it's interacting together. Does that make sense? Okay, so subunits, one of the benefits of having subunits, that tertiary structure, is that you could have this guy could interact with this guy to do one particular function, right? And then later on, this guy could come off, and then later on, it can interact with something else to do another function. So let's say this is to cause your eyes to be blue, right? And then this will allow your eyes to water or something like that. So it can control multiple things by just changing a single subunit. Does that make sense? You look at me like I'm crazy, Alex.
No, I just didn't know that proteins were responsible for so many physiological
Yes.
like responses and just processes and stuff.
Yeah.
I thought it was mainly, the building of tissue and the repair of tissue, et cetera. But you're talking about like physiological responses, like the eyes watering, et cetera. That's just what surprised me.
Yeah. So all of these things are going to be controlled. So the protein plays a huge part. It's a huge part in regulation, huge part in those things. So it's not only involved in tissue buildup or tissue formation, right? It's also involved in that. So in terms of, it can be the adhesive part of the tissues coming together, because tissues are just cells that have been coming together. So what's holding those cells together? We have a combination that it'll be membrane, it'll be sugars, and it'll be proteins. So we have proteins that are responsible for adhesion. So you have glycolipids, right? Glycolipids are sugar fats, right? So glyco meaning sugar, like glycogen or glucose or something like that, glycolipids. Then we have glycoproteins, sugar proteins, holding it together. And then we'll have glycolipid proteins, combination of fats, lipids, and proteins that are coming together, and they are kind of acting as adhesive molecules. Sugar's kind of sticky, so it uses some of that sugar. It also uses aspects of fat to sit there and say, well, fat interacts together too. So we're able to sit there and use different aspects, different properties of these characteristics, different building blocks to sit there and cause things. Okay, so keep talking. How are we doing on time? Not too bad. Okay, so we have enzymes. Enzymes are proteins that basically do work. What does enzymes do again? I think we talked about this last week. I mean, not last week, on Monday.
They're like, what's it called? Catalyst for absorption or something like that, physiological processes or something.
Okay.
I'm sorry.
So ultimately their job is to kind of speed up reactions, right?
Yeah.
Okay, so enzymes are proteins, okay? So they're proteins that are, their job is to basically to get your substrates together so that they'll interact faster, right? So that's part of it, right? Or it can be part of it. But in some way, they basically lower the activation energy so that we can sit there and get our products faster than what we would normally get and not take as much energy. So here's an example of basically how an enzyme works. And this represents the enzyme. This is your active site. Your substrates come into your active site. It allows these two substrates to come together, and then it serves to sit there and allow these products, I mean, your substrate to come together to form its new product. And so it just kind of speeds up that reaction by allowing, in this case, these two substrates coming together to sit there and to form one product. So that is what's happening. We have two substrates coming together, the reaction occurs, and then we get our product made, and then our product is released. And then the enzyme can be reused, okay? So if we have a drug that binds to this particular active site, right? This substrate won't be able to bind. We call this drug A. So if we use drug A, it binds to this particular active site. Substrate 2 is able to bind, but there's no substrate A, so you don't get any of this produced, right? So this guy would be called a competitive inhibitor, right? Because it inhibits this process by competing with one of the substrates. You guys get that? Okay. So then you're not able to sit there and do it. So often a lot of the drugs that we take, like let's say aspirin and things like that, what they're doing, they're going to be a competitive inhibitor or a non-competitive inhibitor, right? And so often they're acting on enzymes to prevent the enzymes from functioning. And so this process, it takes forever for these guys to find each other in our solution. So it's not speeded up by that enzyme. So it doesn't happen as fast as it should. So we get very, very little produced in that case. So often drugs that we take inhibit one of the enzyme's characteristics. Make sense? Okay. The other possibility is called allosteric. So we can have a drug that binds to this that causes this guy to close, right? Now it's not a competitive inhibitor because it's not competing for that spot. It's just binding it and keeping this guy from being open, right? So it kind of closes up. So this guy is no longer going to bind. That is called an allosteric. So you're actually changing the shape, and it's considered a non-competitive inhibitor, right? So it's binding to the enzyme itself, and it causes the enzyme not to function because it's not able to bind to that. Okay. So does that make sense? Okay. Okay. So far we've talked about saccharides. Saccharides are also called carbohydrates. Carbohydrates. We've talked about lipids, which are your fats, your cholesterols, your prostaglandins, and then we've talked about proteins, right? So those are the 3 building blocks so far. Okay, our last building block. Our last building block is nucleotides, okay? Or say our last family is your DNA, your nucleic acids, okay? So your nucleic acid is your last family, and then so we'll talk about building blocks, I should specify. So nucleic acids So these are the nucleic acids or the nucleotides that are found. There are, for RNA, RNA stands for ribonucleic acid, and DNA stands for deoxyribonucleic acid. Okay. So the difference between RNA and DNA is the sugar molecule, right? One of the major differences. There'll be another one here in a second. So deoxyribose basically means that we've gotten rid of one of the oxygens on this 5-ring sugar, okay? 5-carbon sugar, where ribose actually has that oxygen. So here we're lacking this oxygen. By us lacking that one oxygen, right? You see it here? By us lacking that one oxygen, it allows this structure to be more stable, significantly more stable than a ribonucleic acid. Okay, so RNA is less stable than DNA. DNA is more stable. So RNA is going to be serving as a transitional tool, where DNA is kind of like, it's kind of like the template, right? It's the template that's going to stay around forever, for a long, long time. Not forever, but for a long, long time. Where this guy is only going to be around for a little bit of time, okay? And so just by removing that one oxygen allows for more stability of the structures, okay? So DNA, we have adeno, it'll be adenosine triphosphate, right? We have G, we have the C, and we have the T. So we have the adenine, the guanine, the cysteine, and the thymine, okay? So DNA will be composed of these groups. So that's gonna be your nitrous base, right? So we have our sugar, we have our nitrous base, and then we have what we call the phosphate group. So DNA is composed of either an A, a T, a G, or a C. So that's going to be the sequence that we're going to be modifying. So each of your genes is going to be composed of those 4 nucleic acids, right? RNA is going to be your U, your C, your A, and your G, okay? So it's going to have this as your sugar, still going to have the phosphate, and then it's going to use these different ones alternating, okay? Does that make sense? Or did I kind of lose you? You can be honest, because I'll find you.
So thymine, the thymine's like, that's like the deciding factor, right?
Yes. So if we're talking about RNA versus DNA, RNA is going to use C, U, T, C, U, A, and G, RNA. So you're going to have, using my pencil, there we go. So RNA is going to use C, U, A, and G. DNA uses C, T, A, G. Okay, so, and the difference between it, that's one thing. The other thing that's different between the two is that DNA is going to be deoxy. So that means its sugar is going to be a deoxyribose, where RNA is going to be ribose, right? Okay, and so if you look at this, this is showing you, here's your phosphate, right? Your phosphate group. So they all have a phosphate group. They're all going to have a sugar, and they're all going to have a nitrous base. So these are your different nitrous base, okay? So DNA, it forms a double helix, and I'll show you this in the next slide. So we'll have an A, G, T, C, right? And these guys are covalently linked by that phosphate. So they're all connected by that phosphate linker, right? So that phosphate diester linker, that connects that A to that G to that C. This here represents your nitrous base. These are your nitrous base. So that's to distinguish between the nitrous base. Like this would be an A, adenine. So that would be here. So we just say A, G, C, T. These guys are covalently linked to each other. Now DNA forms a double helix. So it interacts by hydrogen bonds. A interacts with T, G interacts with C. Okay, and so these guys are covalently linked to each other. And these guys are interacting by hydrogen bonds. So I'm just going to draw these to represent our hydrogen bonds. Okay. So A interacts to T by hydrogen bonds. So they pair up. And that allows us to sit there and keep those two strands. One strand is used for coding, and the other strand is just kind of used as a template, right? Okay. The other thing about RNA can form double helix, but it doesn't like to form double helix. RNA prefers to be single, like a single strand, not a double strand. Where DNA, in most cases, is going to be a double strand. If it's a single strand, something's wrong. Okay, so DNA, double helix. And I know we're going over this fast. You just kind of need to know some of just the fundamentals. We're not going to get too much into the nitty-gritty, so I'm not going to worry too much about telling you how to decode and all of that stuff too much. So, okay, so as I said, DNA, it has adenine, thymine, guanidine, and cysteine. So these guys go together, right? So here, this here would be your adenine, your thymine, so that's A, T, then this guy would be G. Then if you look on the other side, T pairs up with the A by hydrogen bonds. The T pairs up with the A, the G pairs up with the C. And so these interactions are all by hydrogen bonds. So that double helix is being formed by hydrogen bonds. Okay, and then this here represents that sugar backbone. So it's that sugar phosphate backbone. So it's your sugar and that phosphate group that are connecting those together covalently. These guys are covalently connected. So all of these guys are on one strand, and then these guys are on the other strand that are coming together by hydrogen bonds. Does that make sense? So it's kind of like,
So RNA is going to be single and DNA double.
what was that?
So RNA is going to be single and DNA double.
Yes, so RNA is going to be a single strand. Now it'll fold upon itself, and it'll interact with other parts of its RNA, fold up. But in most cases, RNA is going to be single strand, where DNA is double strand. One strand, this is the other strand. Okay. Does that make sense? Okay. So you guys have heard of ATP, right? So ATP is like cellular energy. So ATP is what we use to sit there and keep our body functioning. So ATP is just, it's that ribose group with 3 phosphates, right? So that allows us to have adenosine triphosphate, or ATP. Adenosine diphosphate, ADP. So that means it only has 2 of the phosphates. Adenosine monophosphate only has 1 of those. Now, all of these guys are able to sit there and generate energy, but ATP generates more energy than ADP. And then often you'll get shuffling between these two. AMP is very weak in energy. It's occasionally used for energy where you'll remove that last phosphate, and then you'll just end up with adenosine, which is just the adenine plus the ribose structure. Okay. You see the distinction between it? This is your adenine is just that's just your nitrous base component, and then adenosine is your nitrous base plus the ribose. So if you look at it, adenine, and then we had that adenosine, so that OS helps us remember, oh yeah, that has that sugar part of it. And then adenosine monophosphate, 1 phosphate, adenosine diphosphate, that's 2 phosphate, adenosine triphosphate.
So does our body use those like in
Is there a certain order?
like
Yeah.
certain,
body
there's a certain
prefers-
hierarchy? Like does it go ATP, and then once ATP starts to get depleted, it works its way down or something?
Yeah, so what happens is that our body starts off with this, right? And so there's certain enzymes that prefer, almost a lot of the enzymes prefer ATP. So then what they'll do, they'll remove that phosphate, they'll break this bond. By breaking that one bond, it releases a lot of energy, right? So the most energy is coming from that single bond. And then we'll have ADP plus inorganic phosphate. So we'll usually just call that PI. So ADP plus PI. So that's what happens after we break that bond. Now, there are certain enzymes that will use, that is capable of using ADP, right? Again, ATP is usually preferred, but there are certain enzymes that are able to use that, and then they'll break that down to AMP, right? All right. And so then there's a little bit of energy in here, right? Not too much, but a little bit. And then AMP can be broken down to just adenosine, right? Just that A, right? So in that case, that adenosine, this guy has very little, to remove this, it has very little energy associated with it, so it hardly ever happens. The only time that is ever done is usually when it's trying to remake it or something like that, or there's something wrong with, let's say, one of the phosphates or something like that. So it's not too common. But each of those provide some energy when those bonds are broken. This guy provides most energy. Breaking of this very first bond has the most energy associated with it. This guy has a little energy associated with it. But these two guys are going to be involved in energy. So that's why they're called high energy bonds. More, this has a little less, this has hardly any. Okay? Questions, concerns, Cash? Okay, how are you feeling? Are you feeling overwhelmed yet?
Just a little bit.
So let me ask you a couple of questions. We'll see how much you guys learned from this chapter so far. Okay, so what are the 4 families of biomolecules?
Nucleotide,
So we have nucleotides. Here, let me find.
saccharides,
Carb,
carbohydrates,
carbohydrates, proteins.
Okay,
and peptides.
okay. So we had
Peptides are, oh, that's right, proteins, peptides, nucleotides. Lipids.
Lipids, yeah.
there you go. So we had nucleotides. So we have peptides. We have lipids. And carbs, carbohydrates. Okay, my peppermint candy falls into which category?
Carbohydrate.
Okay. So let's see. In terms of carbohydrates, what type of carbohydrates do we have that we've talked about?
Monosaccharide, polysaccharide, disaccharide.
Okay, give me an example of a monosaccharide.
Sucrose? No. Fructose.
Okay, so we have fructose. Okay, give me an example of a disaccharide.
Sucrose. Or do you
Well-
mean like more specifically like
Oh,
lactose?
that's right. Sucrose is a very specific example. Yeah. Okay, what about a polysaccharide?
Starch.
Okay. Okay. What about lipids?
Cholesterol.
And
Or
cholesterol is in what family?
The steroid family?
Steroid, that's right. Okay. What else? What other type of lipids do we have in our body?
Saturated fat and unsaturated fat.
So we had those fats, and those fall under which category? You remember?
Phospholipids.
Nope. Try.
That's
Triglyceride.
Triglyceride. Okay. Let's see. Any other?
Phospholipids.
Phospholipids. Oops. Anything else? Starts with the pro.
Bro, I don't think I wrote it down in my notes. I put it prostaglandin.
Yes.
Prostaglandin, yes.
I was like, I don't know that word.
Like prostate.
Prostaglandin.
Prostaglandin. Okay. Okay, let's see. Peptides. So who falls under here?
The aminos.
Amino acids. So that's our building blocks. And then if we put amino acids together, then they form what?
Proteins.
They form our peptides, and then they form proteins or polypeptides that are proteins. Oops. Okay, and then an example of a protein that does work in our body. Starts with an E.
Enzymes.
Okay, so just to kind of talk on proteins a little bit, we also have proteins that are for structure, like your hair, your nails, things like that, right? So, okay. Nucleotides. So what are the two major groups
RNA
of
and
nucleic
DNA.
acids? RNA and DNA. What is the difference between RNA and DNA?
RNA is a single strand and DNA.
And then multi-chain.
Single strand and then
And
double
multi-
strand. Okay.
RNA has the sugar ribose and the DNA
Okay.
has the deoxyribose.
So this guy has ribose. And this guy has deoxyribose. Oxy. Anything else?
Yeah, the thymine thing.
Okay. So we have A, T, G, C for DNA, and then we have A, U, G, C. So in the case of
GUAC, like guac. That's easier to
Guac.
remember.
okay, so I'll remember that. Okay. How you feeling? You feeling pretty good?
Hungry.
I'm hungry too. Okay, so we'll stop here. And for your lab, so your lab this week is going to talk about ionic and covalent bonding. It's another simulation that is on Canvas, and then your lab for Wednesday is going to be your exo lab, your
Physio.
yeah, bio exo.
Okay, so you said the lab for Wednesdays. When does that one due though? You're saying that's
So-
due next Wednesday?
The lab today is due next Tuesday, and then the lab that you guys are doing for today is due next Tuesday, and the lab that's on Monday is next Friday.
Okay.
Okay.
Gotcha.
And then you said we're not going to do a live one until
So-
next week.
So next week. So it'll probably be on Wednesday. So hopefully you guys have your kits. If not, it may be pushed back by until that next Monday. So
Cool.
okay. So is your brain fried yet?
Yeah.
A little bit.
Good. Because I'm doing my job. Okay, so let me stop sharing and I'll see you guys next week. Have a good
Awesome.
weekend.